Welcome to the Derm Club podcast. I am Dr. Hannah Kopelman, and today we have Dr. Animesh Sinha. So I want to start off by talking a little bit about a new topic called pemphigoid or pemphigus. If you have ever had unexplained blisters appearing on your body, pemphigus or pemphigoid could be the culprit. Today, we're going to discuss the intricacies of pemphigus and pemphigoid with Dr. Sinha. Dr. Sinha is the former chair of dermatology at the University of Buffalo in New York, and Dr. Sinha's long-term research interests have been focused on understanding the genetic and immunologic basis of complex skin disorders. He has published over 150 peer-reviewed articles, including 4 in the Journal of Science. Dr. Sinha, welcome to the Derm Club podcast.
Very nice to be here. Thank you.
So I want to start off by having you just tell us about your career track, what you're doing now, and how you got there. I think this would be a good way to introduce yourself and to walk us through the topic of today.
Sure. Well, I entered medical school and was interested in learning about, the human body and disease. And this journey to where I've come now actually started particularly in second year medical school when I had my first lecture in immunology. And I really found it a fascinating subject, understanding how the immune system deals with a conundrum that still is one of the most basic mysteries in science, is how does the body distinguish self from non-self? How do we have an immune system that's so diverse and so adaptive and can react to all this universe of pathogens and maybe cancer cells and abnormal things, but stay neutral or tolerant to our own self tissues. And that's the crux of self non-self tolerance. So I was fascinated by this in medical school, and so much so that I pursued a PhD in immunology after medical school. Went on to do postdoctoral work in autoimmunity, and which is the flip side of self-tolerance. When you lose self-tolerance and the immune system starts to attack your own tissues. And that became a lifelong pursuit and the thrust of my career. I then went on ultimately to train clinically in dermatology, but I kept my research interests going throughout medical school, fellowship, residency, and then through my faculty appointments at various institutions. So I've had a long interest in the immune system, how it functions, how it breaks down, and why autoimmune diseases happen. And I want to point out that this understanding of autoimmune disease is very relevant because autoimmunity collectively is about the second or third most leading cause of morbidity and mortality, and is a huge health and economic burden to the tune of over $100 billion per year as a group. So there are a lot of commonalities across autoimmune disease, which can occur in any organ system, including the skin. So although we study, and my work has been focused on autoimmunity in the skin, and particularly the blistering disorders like pemphigus and pemphigoid, we really address issues about, through studying these diseases, we address issues more broadly about the mechanisms of autoimmunity and how to ultimately unravel those mechanisms to come up with ways to better manage and treat patients and to understand the workings of the immune system. So that's been the arc of my career and led me to pursue this line of work to really try and understand one of the most complex systems that we have, which is the immune system.
Okay, thank you so much for sharing that. So we start off with a very basic question, but a confusing question to some. What is the difference between pemphigus and pemphigoid? I know these terms are often thrown around and people don't realize that there is a difference between them. So if you could just start off by explaining that to us, I think it would be a great help.
Sure. So as I mentioned, that autoimmunity can affect any organ system, including the skin. And there are a number of autoimmune diseases in the skin, including alopecia areata, where you lose your hair in clumps, vitiligo, where you can lose your pigment, cutaneous lupus. Psoriasis has some autoimmune and immune mechanisms involved. And also the blistering disorders or autoimmune blistering disorders. And the autoimmune blistering disorder group can be divided into two basic categories. One is the pemphigus group, and the second is the pemphigoid group. And the differences are that in the pemphigus group, the split or the blister occurs within the epidermis, the top layer of the skin, and actually just above the basal layer or the bottom layer of keratinocytes that comprise the upper layer of the skin, the epidermis. In the pemphigoid group, the blistering occurs just below the epidermis, right at the epidermal dermis junction and the dermal epidermal junction, and this results in a slightly different clinical presentation because the split or the blistering occurs a little lower in the skin in the pemphigoid group, that the blisters tend to be a little more tense, thicker, where in the pemphigus group, the blistering is a little higher in the skin, in the epidermis, and often the blisters are more friable, sheared off. Often you don't even see intact blisters, just a shearing and erosion of the skin and those lesions. In the pemphigus group, the prototypical disease is pemphigus vulgaris or PV, but there are other variants as well. And in the pemphigoid group, the poster child for that is bullous pemphigoid or BP, but there are variants of that too. But the main distinguishing factor, as I said, is the level of the split in the pemphigus group in the epidermis versus the pemphigoid group, which is just below the epidermis at the dermal epidermal junction. And the reason for these is because the autoantibodies, they're the mediators of disease ultimately, are targeting slightly different proteins that are important to the integrity of the skin, and therefore, and the location of those proteins varies anatomically, and so that determines the level of the split, and that's the main distinguisher between the pemphigus and the pemphigoid groups.
Very interesting. So I understand you mentioned some other autoimmune conditions. Is there a specific association of autoimmune conditions with pemphigus versus pemphigoid, or are they typically both associated with the same autoimmune conditions?
Well, you bring up an excellent point. So let me pull back. So all autoimmune disease can be classified as complex in etiology. That means they're multifactorial in origin, meaning they're both environmental and genetic factors, most of which we do not know yet, that conspire together to dysregulate the immune system and cause autoimmunity. So the question is that how there is a genetic basis, and that there's probably some commonalities between autoimmune disease. And what we do see is that if you have one autoimmune disease, if an individual has one autoimmune disease, there is a slight increased risk of having another autoimmune disease, and in themselves or in their family members, in their first, second, and third degree relatives. And so that suggests a common cause and maybe even common genetic risk elements. And it is not always random which autoimmune diseases are associated with another given disease. So our group has published that there are disease clusters, certain autoimmune diseases that are even more likely to run together in an individual with a particular autoimmune disease or in their family members than left to by chance. But there's still a lot of work to be done about that. In Pemphigus vulgaris, PV, we've identified a disease cluster between pemphigus, rheumatoid arthritis, and type 1 diabetes and autoimmune thyroid disease. So there's a slight overall increased risk of any autoimmune disease in an individual who has a disease, an autoimmune disease, and in their family members, and a little more likely in some of these diseases that I just mentioned that cluster together. So the risk is distributed through a genetic mechanism, but remember that that the diseases, these diseases are not just multifactorial, but they're polygenic. In other words, there are multiple genes involved, most of which we don't know. And there's probably some overlap in the genetic risk elements between certain autoimmune disease, so that they tend to run together a bit.
You mentioned that there's this genetic component. Does that mean that if I have it, say, then I'll give it to my offspring? Or is that not always the case?
So, so the key thing to remember here, that there is not a single gene or a single gene defect that's involved. There are many diseases that, like cystic fibrosis or something, that where it's really a single gene that's involved, and that can be passed down and have a higher risk in offspring and relatives. In these complex diseases, as I mentioned, they are polygenic, so there are multiple genes, and most of which we don't know. So you need a combination of genes and to have the risk factors to give an individual the susceptibility for a given autoimmune disease. And I want to talk about that a little bit, because most of these genes we don't know, but we do know that the human leukocyte antigen genes, or the HLA genes in human, which are key genes and proteins involved in directing the immune response, they are linked to various autoimmune disease. So there's a couple points here. These HLA genes are very polymorphic, meaning that there are many variants or many alleles out there in the population. And that is probably because we want to have slightly different genes at the HLA to, as a population level, have a slightly more expansive and diverse immune system. So a given pathogen can't just wipe out the whole species. But we also started to find out a few decades ago that certain alleles or variants of the HLA genes and molecules are linked to certain autoimmune disease. And in fact, many years ago when I was at Stanford, I sequenced some of these HLA alleles in Pemphigus patients and found there was a very strong association with a couple of these variants. And it gets a little more complicated than that, but it indicates that the immune system is central not only to driving normal immune responses, but also to activating abnormal immune responses. And so that these are key risk elements, but they are necessary but not sufficient. There are other genes that are responsible. It's been very hard to find out what these other genes are across autoimmune disease, because there are probably multiple genes with minor effects. And we are still, as a field, trying to uncover these genes and how they might all work together. I want to make one other point. Although the genetic risk elements, including the HLA molecules, are very important in conferring risk, twin studies indicate that genetic risk only accounts for 20 to 50% of the susceptibility to autoimmunity. The other percentage, or the high, more than half of risk, is related to environmental factors. And I mean environmental factors more broadly in terms of your one's exposure to various pathogens and viruses and bacteria, et cetera, that have shaped your immune system, other toxins, medications, and perhaps in some cases, things that are ingested. These things are even less clear. There have been a lot of anecdotal reports or case reports about various environmental factors that trigger autoimmunity. And we do know that there are triggers of autoimmunity, but the exact environmental factors are not well worked out. So it really does indicate the complex nature of these conditions, multiple genetic risk elements. HLA is very important, but it's only one. It's the prominent one, but it's one of many. And then multiple environmental factors. So it's a really tough puzzle to put together, but investigators around the world over time have been working on these things, and every year we get a little closer to filling in the pieces to this complex puzzle.
So it seems like epigenetics definitely play a role in the development of this disease or other autoimmune diseases. Do we know at this time, I often have patients who come in and they say, should I eliminate this from my diet? Should I eat this? What is it less inflammatory? Does food, or at this point, do we know if any foods play a role?
So there's been a lot of reports and suggestions about various foods that may influence susceptibility or the course of autoimmune disease. It's not clear. There are no strong associations, but there have been some reports in pemphigus that groups with certain foods with alum group that have thiol molecules, or urushiol groups that have phenol molecules in them, could contribute to disease. And in the alum group, we're talking about onions and leeks, shallots, garlic, and the urushiol group, we're talking about mangoes and cashews. Now, in some cases, it just could be the irritation of onions and garlic on open wounds in the oral mucosa where pemphigus lesions often begin. But it also could be that the molecular composition of foods that have these certain chemical groups could trigger the immune system and cross the pathways that lead to autoimmunity. So that sort of pathways leading to disease, from certain foods through the immune system through to clinical presentation, has been suggested but not well worked out, but it's possible. In a similar way, certain medications have been linked to the development of autoimmunity, including pemphigus. Drugs with a thiol group or a phenol group, again, penicillamine, captopril, have been suggested to trigger autoimmunity. Again, the mechanism isn't entirely clear, but it could be that they interact with the immune system and activate it in a similar way that crosses wires with what would happen in pemphigus and ultimately leading to disease. So these are very interesting but complex sort of questions and mechanisms to work out. But as you can see, that there's a lot of interactions between the immune system and the environment and the genetics that shape all of that. So again, and there will be some commonalities across all autoimmune disease, within an autoimmune disease, but there will be some individual differences too for the exact pathways that lead to disease in each patient.
So, there are many other skin conditions that present as blistering skin diseases. When a patient comes in to see you, how do you differentiate what they have, since there are so many differential diagnoses?
Yes, blisters can be for many reasons. So of course, the diagnosis, we go through a stepwise sort of algorithm, if you will, to pin down the diagnosis. We look at the clinical presentation. Where are the lesions? Are they tense? Are they, very friable and thin lesions? Where are they distributed in the oral mucosa or other mucosal surfaces or non-mucosal surfaces? We take a history, were there any family history of disease or other blistering conditions? Was there any trauma or, et cetera, for blisters? And then we usually do a biopsy, and we can do a couple different biopsies, one for regular H&E staining, the normal biology, and we look for histopathology that will show that there is a split in the skin and where that's located, and there's characteristic histopathological features that would point us in the direction of pemphigus or pemphigoid. There are a couple of special pathological tests that you can do to separate the skin layers to see where the antibodies might be binding. So we can do a direct immunofluorescence and see that there are immunoreactants that light up in the skin. That would tell us that there could be antibodies that are causing a problem that are normally not present. And then we can do special types of stains to figure out where exactly that binding is to help us differentiate between different autoimmune blistering disorders. We can do an indirect immunofluorescence, and that's where we take some blood and take the serum and incubate that on substrates such as a different type of epithelium from other species to see if there are antibodies in the serum of a potential patient that are found to bind to the surface of the keratinocytes, and we can light these up with a fluorescent marker so we can see them, and that's called indirect immunofluorescence. We can also take the serum from a potential patient and investigate if it has specific antibodies or autoantibodies to key proteins that we know are targeted in pemphigus or pemphigoid. And we do this usually using a technique called ELISA, where we put the protein that we know is important in pemphigus or pemphigoid, and put them on a plate, a tissue culture plate. And we then incubate the individual serum. And if there is a autoantibody that recognizes that protein on the plate that's been linked to disease, then it will light up, and we can again say, oh, this person has antibodies that target this protein that's associated with pemphigus or this protein that's associated with pemphigoid. For investigational purposes, it's not done routinely, but in our lab, we also check for the HLA molecules. We do a quick gene sequence of patients, and that may help us give a clue that if they type for one of the commonly associated genes that have been linked to pemphigus, then that gives us an additional piece of information that might help seal the diagnosis. So there's a multi sort of layered approach to confirming diagnosis, but the keys are the clinical presentation, and then the histology and the immunofluorescent histology. I will say that it is not often easy to make the diagnosis, and our own group's data, and I think it's corroborated by other people's data, is that it takes 12 to 13 months on average to get the correct diagnosis to pemphigus or pemphigoid, especially pemphigus, from the initial symptoms. And so often patients will go to various doctors. These conditions are by definition rare. So not everybody is well versed on these diseases and how they present. And as you said, there's many different ways you can get blisters. So they often present in the mouth, so they can be confusing and sometimes they're detected first by dentists. So I think the average is 12 to 13 months before a correct diagnosis from time of initial presentation. And the average patient has gone through 5 or 7 different health professionals before they get a correct diagnosis. So these aren't always easy conditions to identify and classify. But we do have tests that can be done systematically to pin down the diagnosis.
So is there any advice, because does it matter to the patient? I mean, of course it matters to the patient. Like very frustrating. They're going to many doctors for different opinions, trying many different treatments with no success because they're wrongly or misdiagnosed because of the lack of knowledge from the clinician about the specific disease. So is there any one piece of advice that you could share with a clinician, maybe that would help you, dermatologists, and the patient, so that they could be diagnosed earlier on?
Yes, I mean, I think blisters on the skin or in the mucosa, especially the oral mucosa, need to be taken very seriously. I would advise a patient or primary care person who sees a blister is to, get them referred to a dermatologist, and particularly a dermatologist with experience with complex medical disease, and maybe even a medical center that is, able to handle these complex diseases. That is a big issue, is that not everybody lives near a major medical center, so access is an issue. Awareness. So there are groups like the International Pemphigus and Pemphigoid Foundation, the IPPF, that have programs in outreach and awareness to patients and to dentists and primary care physicians, to make them aware of the disease, its presentation, and the resources and centers that could handle these diseases in the most timely and appropriate way.
Thank you. Do you ever see a patient who comes in, clinically presents with pemphigus or pemphigoid, but they don't have any of the genetic markers? They don't have HLA, they don't have autoantibodies, but they do in fact clinically have pemphigus or pemphigoid.
So there are, rare cases. Sometimes people don't have the two common HLA alleles that are most strongly associated with pemphigus. And in fact, that's a very complex story. There are different ethnic populations and worldwide populations that have slightly different HLA associations with pemphigus, but somehow arrive at the same disease pathways and get disease. And so that's still being investigated. There are patients that don't have the classical autoantibodies to that are directed to desmoglein 3 and desmoglein 1, which are key proteins in helping skin cells stay attached to each other, or in the case of bullous pemphigoid, BP antigen 180 and BP antigen 230. So you do see sometimes patients that don't have those antibodies. They may have other autoantibodies that our group and others are starting to realize can be present in patients with autoimmune bullous disorders. But a lot of the breadth of the immune response and the genetic susceptibility risk alleles are still being worked out. And then from that, we need to not only identify different scenarios genetically or immunologically that could still be associated with disease, but how those different scenarios all link somehow into the pathway to cause disease. So that there are a number of investigative things that still need to be worked out to understand more broadly how disease is happening.
Can you share with us the pathophysiology that leads to this loss of cell-to-cell adhesion?
Sure. So as I mentioned, all autoimmune diseases are complex. There are genetic and environmental factors that are very relevant, and they work together. We are still working on identifying the full set of genetic and environmental factors that might be involved in the disease and within a given patient. But we do have a basic roadmap to how autoimmune bullous diseases, and especially pemphigus vulgaris, happen. So I'll take you through the most typical scenario. You have to have genetic risk element, in particular key HLA genes and proteins that activate T cells that have an autoreactive potential. Now, what these HLA molecules, they present certain bits of protein or epitopes to T cells, T lymphocytes, a subset of white blood cells that are key players in the immune response, but they activate these T cells. And in this case, they would activate T cells that are reactive against the self protein. But we're still not exactly sure which bits of protein activate these T cells. Could be a cross reaction with viruses or bacteria in some cases. And anyways, these T cells ultimately get activated. And these activated T cells are required to promote the activation of B lymphocytes, another class of white blood cells, and that produce antibodies, these white Y-shaped proteins that sit on the surface of B cells. And when they get activated, they can secrete these Y-shaped proteins in the circulation. And these are antibodies, and if they're directed against self-proteins like desmoglein 3 or 1 in pemphigus, or bullous antigen 180 and 230 in bullous pemphigoid, if these B cells secrete those proteins, those antibodies, then those antibodies will then go through the bloodstream, enter the skin, and bind to key components in the skin that are relevant for the integrity of the skin and holding the skin cells together. And once they attach, then that ultimately leads to the dissolution of these bonds that help the skin, that are required for the skin to stay together, and you get blistering. Exactly how these antibodies cause this cell-cell dissociation, or the term is acantholysis, how that exactly happens is still a matter of intense study. Could it just be that the antibodies bind and they just physically disrupt these bonds? That's one scenario, but it could also be that additionally that the binding of these autoantibodies to these important cell-cell adhesion proteins activates something internally in the cell that further leads to some signaling pathways that are relevant ultimately to cause the cells to fall apart. So we are, and others are, investigating all aspects of this roadmap, from the HLA genetics, the activation of T cells, the activation of autoreactive B cells, cytokines, and other immune pathways that are relevant for the propagation of the autoimmune response, and ultimately how the autoantibodies cause acantholysis or cell-cell dissociation once they bind. And these are all active areas of investigation. And why that's important is, one, we want to understand, we need to understand these details so then we can come up with hopefully better and more targeted therapies. So there could be a whole chain of events that are important. But if we understand the links of the chain, and we can block even one of the links in the chain, or cut it and block that that pathway, then we may have ways to block this autoimmune pathway and help patients escape disease. So these are all areas of very active investigation. We've made a lot of progress over the last 2 or 3 decades, and the increase and the work continues, and we still have work to do, of course, but we have come a long ways. But there's the immune system and the body are very complex and not easy to reveal their secrets, but people are working on it.
Science is evolving and you're a part of it. So what is the first line treatment for pemphigus or pemphigoid?
Sure. Well, because these are autoimmune diseases, there is a misdirected autoimmune immune response that targets the wrong proteins, if you will. And so the basic thing is to suppress that immune system. You can do this generally by blocking the entire immune response, and prednisone or steroids by a variety of mechanisms can suppress the immune response, and it's very effective. So you knock down the entire immune system, and you can, now you block your pathway to autoimmunity. The problem is that you've knocked down your whole immune system, and that's not necessarily good. So you've done it in a non-specific way. And prednisone, again, for a variety of reasons and through various mechanisms, has a lot of side effects. And so It's not an ideal drug by any stretch to be on for a long time. Some people can't tolerate it at all. It is very effective. But our basic sort of therapeutic ladder for treatment of these diseases is to get them on a relatively high dose of prednisone to knock the immune system down. And usually we start at 1 to 1.5 milligrams per kilogram per day. And that usually works out to about 60 to 80 milligrams per day. And that will continue until the control of disease phase, which is when new lesions cease to erupt, and there's a beginning of the healing of lesions. And that's usually, it varies, but it can be for 4 to 6 to 8 weeks. And then we can continue or slightly taper, usually continue on through the consolidation phase of which, is usually takes up to 12 to 8 to 12 weeks. And that is when there are no new lesions developing for the past 2 weeks, and there is a healing of the majority of lesions, say even 80% of the lesions. And then we can start to taper down the prednisone. Now, during this time, we may or may not add immunosuppressive drugs, non-steroidal immunosuppressives that, again, work by a variety of mechanisms to block immune cell proliferation mostly. And they are also very useful, but again, they also are knocking down the entire immune system. We have other options, including intravenous immunoglobulin, which is a pooled collection of immunoglobulins or gamma globulins in the serum that for some reason, for reasons, again, we're not entirely sure, we can flood the system with immunoglobulins, and that seems to sort of drown out the offending autoantibodies that a patient might have. We have now some more targeted therapies like rituximab, which is a antibody that's been engineered to target certain key proteins on B cells. CD20 is its target on B cells. B lymphocytes, remember, are the subclass of white blood cells and lymphocytes that secrete the antibodies or autoantibodies that are relevant for autoimmune disease. So if you knock down the B cells, you could imagine that now you're knocking down that engine that drives the production of the autoantibodies. And that's been relatively a new development in the last 5-10 years, and it has been actually very useful in Pemphigus vulgaris. Again, you're knocking down, though, a whole arm of your immune system, and though while it's been fairly safe and fairly effective in many, not all patients, we still ultimately want to get to a place, I think, where we want to be much more targeted and surgically precise, if you will, in terms of our therapeutic approach. So if we could knock down not the whole immune system, not just the T cells that are relevant to disease, or not just the B cells that are producing the autoantibodies, but if we could target only those B cells or only those antibodies that are causing disease, only the ones that are specific for the key proteins that are relevant, in disease development, like desmoglein 3 and 1 in Pemphigus, both pemphigoid antigens 230 and 180 in both pemphigoid. If we could just target surgically those aberrant clones or autoantibodies, then we would have I think a more effective and more safe, ultimately, in terms of side effect profile, more a therapeutic approach. But that's complex because we still need to find the exact mechanisms that remember, there are probably some redundancies in the specificities of the T and B cells and autoantibodies that cause disease, even though we have identified in many cases some of the main drivers, but there are probably others. So it's a work in progress, and then to develop the actual therapeutic agents that can be that specific is, takes work and time, but people are making progress on this front.
Very exciting. So are you doing any current research with future treatments, any work with the Bruton tyrosine kinase inhibitor, which you can explain to our audience how that works, or also I know there's some treatments being used to infuse antibodies against the FcRn receptor to increase the removal of autoantibodies, which would help us eliminate pemphigus or pemphigoid. So
Sure.
I'm just curious where you are in your research, and if you could share that with our audience.
Yes, of course. So yes, there are a couple other approaches that you just mentioned. There is there there have been a couple trials, one ongoing now on phase 3 trial on targeting the FcRn receptor as a therapy for pemphigus, and then I think they're going to try it in bullous pemphigoid as well. And this is a receptor, the FcRn receptor, that is an important molecule in sort of the maintaining antibodies in circulation. So they kind of autoantibodies, so these Y-shaped proteins, antibodies are, and they bind to this FcRn receptor. They're sort of recycled and sort of protected and then put back in circulation. And the thinking is, the rationale is, if that FcRn receptor molecule could be targeted and blocked, then it wouldn't be able to function in its normal capacity to protect and recycle antibodies, and therefore also within that group, the autoantibodies. And so a couple of different companies have developed drugs on that basis, and they're in clinical trials. There's some favorable results that have come out, and it has to be determined if they'll hold up, if the results will hold up, and safety profiles will hold up, and if they will be ultimately approved for this indication. So, but that's a promising lead. BTK, you mentioned, is a molecule that's involved in B cell activation and function. And again, B cells are important to make the antibodies or autoantibodies. They're relevant to affecting disease. And so there was a trial recently with an inhibitor of BTK. It was Principia. I think Sanofi bought them, but I think their phase 3 trial results were not positive. Their phase 2 trial results showed some favorable results. So I think that's been shelved, but doesn't mean that there won't be other approaches, with similar approaches, maybe with different, antibodies that could also work off that pathway. So there are a number of new things being tried, and I think in the end it may be that we need a combination of these things, because not every patient will respond to every medication. We still have a ways to go to match therapy with therapeutic response. We don't know which patients are going to respond to a given medication. And again, that may be based on their genetics, their particular pathway of disease, and the intricacies of their autoimmune response. So I think that will be the next frontier will be going into more antigen-specific targeted therapies. So going beyond general immunosuppression or somewhat targeted therapies like rituximab and FcRn inhibitors, BTK inhibitors, to very antigen-specific approaches where we try and knock out just the offending T and B cell clones or autoantibodies that target the key proteins relevant for disease in a particular condition or patient. And then the final frontier, I think, will be individualizing these approaches to given patients. So while there may be a general definition for disease, what we're seeing is that there's a lot of variability and heterogeneity within the complexity of the genetics and the immune response in any given patient. So it may be that ultimately different combinations of therapies and ultimately very, very individualized targeted therapies will be the most effective. And, I think that will take some time, but that's the path that we're on.
So, this is an autoimmune condition. Do patients have to have a concern for recurrence? And if so, are there maintenance therapies?
So autoimmune disease, these are chronic diseases. There's not a single autoimmune disease for which we have a cure. Uh, the natural course of these diseases is to wax and wane. I think that probably reflects the push and pull of the internally of the immune system. I think you have got, it goes sort of off track, one's immune system, and then there are countermeasures built into the immune system to try and pull back on that, put the brakes on that. Medications, I think, can help tip the balance towards putting the brakes on. So, but in typically autoimmune disease wax and wane, they go through the cycles of flares and remissions. In the bullous disorders, they tend to, though, get the flares get less frequent over time. We don't know. Again, maybe the autoimmune response peters out, or the internal brakes, with or without the help of medications, help to, ultimately tamp down the response. But it is an ongoing struggle, and to achieve long-term remission, I think we're starting to get better results with the medications that we have now than we did 10, 20 years ago. But we're still working that out, and that will depend on our better understanding and elucidation of the complexities of the genetics and immune response in a given disease and in a given patient.
Hopefully the targeted therapies will be, a possibility that will be a longer term control for this disease.
Yes,
That would be
that's the
very
holy grail.
exciting. Uh, so I just want to ask you a few more questions before we say goodbye, but have you seen any association with a flare in pemphigus or pemphigoid with hormonal changes or pregnancy?
Yes, that's complex. I think the answer is yes. I can't give you exact direct correlations, but there's a couple of interesting things here. First of all, autoimmune diseases tend to be more represented in females and males. In pemphigus, it's about 2 to 1 females to male. Across the globe, most studies show that. In some autoimmune disease like lupus, it's 9 to 1 females. And so we don't know the basis of that entirely. There are a number of different hypotheses for that. How pregnancy and hormones affect a patient's condition when they have disease, it can often exacerbate it. But in psoriasis, I remember, a third of the patients got flared when they were pregnant, a third got better, a third were neutral. In pemphigus, I don't know if we have enough data on that, but there's no doubt of the influence of sort of these hormonal effects do interact and sort of probably sway the immune system. How that eventually is manifest in a given patient, I think it's hard to predict.
Okay, well, I think there's a lot to come in the future. And Dr. Sinha, thank you for sharing your depth of knowledge with us. I have a greater understanding after our conversation. If there's anything else we didn't cover today, or if you have any questions for me or Dr. Sinha, please feel free to reach out. And thank you for joining the Derm Club podcast today, Dr. Sinha.
Thank you very much. My pleasure.
I hope you enjoyed this episode of the Derm Club podcast. If you found the discussion today to be valuable, please subscribe and share. I look forward to seeing you in the next episode as we continue to delve into dermatology and skincare with the world experts.